Now we see it. Thank you. So please start.
Yeah, thank you. So my talk is about how mathematics and AI are revolutionized the bioscience. Yeah, I'm Guo-Wei Wei. In this community, in this context, in this conference, I should say I'm a biologist. Otherwise, I would say I'm a mathematician. So my talk is in the between. There's not much math in my talk. The whole thing is designed for people in the biology. They can somehow understand what I try to talk about. Yeah. So everybody knows at this point, we're in a very sad situation. We have a pandemic, so we have a lot of casualty over there. And the question is, what can mathematics do for it? And how, and the other question is for biologists still, for other people, everybody concerned is how will the COVID unfold in the future? So what is going to happen? So scientifically, so like variants, what type of variants are going to happen? So my talk is to try to design to answer those two questions. So let's start it as a little bit of things about AI. I mean, AI, people in the biology community, they don't pay too much attention to AI, particularly in the US. A few years ago, until something so-called AlphaFold, I mean, that stuff basically shocked everybody by when the protein folding competition, that's the first one, was December 2008. And then actually last year, they win again with a huge win, like 90% since something like that, they win everything. So, I mean, the idea is to try to, from sequence to fold of a protein and to see what is going on for the protein structure. So that's the AI basically win everything over there at this point. So, but when you try to use AI for biology in general, for example, if you want to do drug discovery, you still face a problem. Is a dimension is extremely high, it's 3n. N is about 5,000 for average protein, things like that. And also machine learning dimension, if you just take a shot here, from here to here, use 1,000 pixel, and you are getting to a dimension of this one, and times m. M is a different type of atoms over there. So, and also you have a different size. So from a machine learning point of view, it's not scalable. And also you have a complexity, all kinds of different interactions over there. So what's the solution for that? There are two schools of thinking. And so if you say a protein has an n atom, n is about 5,000, and each atom has n electrons, that's a little n, it's roughly about 10. So if you use like a PDE type approach, Poisson-Boltzmann, PMP, you are in the three-dimensional space, okay? If you want to do multi-scale, yeah, in the dimension is higher than 3D, but it's smaller than 3n, n is about 5,000. If you do molecular dynamics, you are about 3n, so n is 5,000. So, and then if you do quantum mechanics, so basically mission impossible. So on the other hand, if you think about a different way, instead of this way, you say, okay, look at the solar system. We have about all kinds of different planets over there. We're not interested in most of them. We're only interested in the surface of this blue guy. That's Earth. So we look at a differentiable surface and a differential manifold. We get into 2D. And the hypothesis say the intrinsic physics lies in the low dimensional manifold, embedded in the high dimensional space. We wanted to find out what is that. And we can do different mathematics. Differential geometry is one thing, algebraic topology, and you can do graphic theory, you can do index theory, whatever, so on and so forth, all kinds of different ways. And then you do dimension reduction. So, and the question is, can this method work? So this is the answer. Let's show you how we do things. Our strategy say similar to other people, we do all kinds of biology, sequence structure, bioinformatics, systems biology, and also like other people doing, we do all kinds of machine learning, all kinds of different machine learning technique. Apart from that, we are unique in terms of we use algebraic topology, differential geometry, and graph theory combined with those three. And we certainly make use of the achievement in the mathematics, include the algebraic topology community we are talking about here. So this is a contribute a lot to what we are doing. So let's talk a little bit about the topology. I mean, these things I tried to talk these things to the biologists, so that's why I include things this one. A little bit of things I wanted to mention about the Nobel Prize in 2016 in physics is given to low-dimensional topology. So and this is going on have a bigger impact in the science. So try to say what is really important for topology. Certainly, everybody here knows a lot of things, but typically most important things are the topological invariant and all kinds of different topological invariants over there, include Klein index and things like that. But the Betti number is something we talk very often. So Betti 0 is the number of connected components. Betti 1 is the number of circles, and Betti 2 is the cavity. So, I mean, this is just roughly say is not a rigorous way to say that. And try to give one example because it is community everybody knows for torus, and then your Betti 0 is 1, and the Betti 1 is 2 because you can see two rings over there. And then you have a Betti 2, everything inside is the input. That's why I have Betti 2 is 1. So this is the basic way, but I realize that this is a continuous subject. If you really want to do protein, you have to use simplicial geometry. You see, you have a set of points like atoms over there. So in a protein, well, in a molecular, in that case, we need a different language. And so we have a simplest 0, 1, 2, 3 simplest, things like that. And then for a given set of atoms over there, in order to identify those simplest, you have to give a cutoff. Say this is the radius over there, yeah, with a different radius, and then you got a simplest over there, simplicial complex. And what do we do with these things? We use that type of technique that has been developed in the community for the past 20 years. So you take a simplest and then form a chain group, and use a boundary operator to generate other groups like circular and boundary group. And then we have a homology group. From there, just look at the dimension of homology group. And then we got the Betti number, the topological invariant. And then if we do a filtration process, we get a persistent homology. There's a whole lot of people here contributed to these things. So this is still ongoing for the community. So we try to say, okay, if you have that, or try to illustrate it to biologists, so everybody in the room probably don't need this. So basically you have set of points over there. You can see if you have a filtration, you have a simulation complex come up here, and then so you can identify the Betti number, things like that. Every time something they are connected to each other, you reduce the number of independent components. That's why the bus doesn't go any further. So, and then in a few minutes, in a moment is going to have something, have a cycle over there. You can see things are connected together. So you got this one, and then you see the Betti one come up, and then you get other Betti one come up. And then if the radius keep increasing, this one is going to be killed. And then you see this Betti. So this Betti one is going to stop moving at the point that this one, so it's going to fail. So that's it. So you see. So what we got from here? So at a few minutes we got everything. So and then we got a pattern, and this pattern is a topological fingerprint. So it's a topological fingerprint for this set of points. If this set of points is a protein, so that means I have a topological fingerprint for the protein. So that's the idea. So if that, for example, this is a protein alpha helix atoms over there with 4, we have a Betti 0. It's this one, we have 4 lines. Finally, if the radius is increased enough, and then it stops at this point, it become 1. And this is 4 at a certain points of the filtration, it get a cycle over there. So we got a Betti 1. So the same way we increase 1 atoms over there, and then we see 1 more line, and also there are upper 4 and the lower 4. In this way, we actually have a systematic procedure to identify arbitrarily complex biomolecules like a protein, beta barrier, and even some data from electro from electron density. You can see the microtubule electron density. We can still do that, use cubical homology. So this is things we did quite some times ago, yeah. So, and then if you want to understand the protein folding, try to represent it in a much cheaper way, this is a way to do it. I mean, this is definitely, you have a two-dimensional representation of protein folding. Otherwise, this is a huge dimension. If you think about it, this is 5,000 atoms over there. So it's 15,000 dimensions past time. So we put the whole thing here into such a, like a two-dimensional representation. So that's the idea, to reduce the dimensionality dramatically for complicated biological problems, such that we try to find out what is the physical law or governing laws over there. So that's the idea. So, and certainly we also have something so-called persistent cohomology. The basic idea is to incorporate long geometric information into the topology. So for example, if I show you like this one, I have a different color, means different type of atoms over there. And the different type of atoms I can put into the homology representation. Those are long geometric information, and then my barcode become colorful. And certainly I have to use some more fancy things like Wasserstein, those type of representation in order to describe what we got. So it's more than just persistent homology. Next, I want to talk a little bit about differential geometry. There's a big expert here. Yeah, so just wanted to say this is also an ancient subject over there, but it has all kinds of different things. If you observe the planetary system, virus and bacteria, they're all like this type of shape. Why is that the case? The reason is minimize the surface. If they minimize the surface, they become more stable. So that's maximize the stability. So that's the basic idea. So how to do that mathematically? To do that, if you take something like that, this is a drop of oil, and you say define a function inside the oil is 1 and outside is 0, and the interface is between 0 and 1. And then you take this function, take a gradient of this function and take absolute value. This is the geometry Maria theory tells us this will be associated with the error. And with this type of error, you times the surface tension, just give you a surface energy. And you do the variation for these things, what you have is the mean curvature. And then you can extend the procedure to turn a mean curvature into a parabolic equation. So by this procedure, you got a mean curvature flow. And from there, we do all kinds of different surface representation, like a DNA, protein, C60, and also virus. So this is something we have been doing this like a long time ago. And in this way, we can go further, try to characterize, use different Gauss curvature, mean curvature, maximum, minimum, all kinds of different curvatures to represent the proteins like HIV receptor, things like that, and some other things like virus here. So recently we did a little bit more on this along this line, differential geometry. We look at the de Rham-Hodge theory and Hodge decomposition. This is the Hodge decomposition. A vector field can be decomposed into harmonic components, curve-free and divergent-free. And for doing that, there's a lot of application in the physics quantum field theory, for example, Maxwell's equation, and it's getting into different forms. So we have a 0 form, 1 form, things like that. And then we apply these things to biology, like cryo-EM imaging things there, and look at the electrical fields distribution, how they get decomposed. So that's the things we try to do. And also, so basically, if I think about what I have done so far, we talk about algebraic topology, right? And we also talk about filtration, multi-scale analysis. So in the between of algebraic topology and multi-scale analysis, we get a persistent homology, right? And then we talk about differential geometry. And in the between of differential geometry and algebraic topology, we really have something roughly of a de Rham-Hodge theory. And then between the differential geometry and the multi-scale analysis of a manifold convergence, things like that. A whole bunch of people are doing that. So the question is, what's in the between of these three different fields. Okay, what is here? So what we try to do is say, okay, look at the big picture. What is here? So we call evolutionary de Rham-Hodge series. This is something we did recently. So what is about? And basically, you have a manifold evolution. So you have a lot of different manifolds over there. And then from a manifold, also incorporate this into the de Rham complex. So, and then you have a different manifold from M0 to M1, M2. And from this one is you have a differential form. So it's M0 with a differential 1 form. This is 0 form 2 form. I mean, that's what it's about. So what is good about this method? So the things good about this method, you see, look at the topology. The topology, they have a problem there, you see. They doesn't describe things here. So you go from this one, this is a homological invariant. So it doesn't describe things getting fat, right? If I'm eat too much, I'm getting fat, the topology doesn't work for that. So you use but the topology, it works for things you have a topological change. So evolutionary de Rham-Hodge theory is able to describe two types of things, both topological change and a non-topological change, and describe a non-topological change in terms of frequency. So this is the Laplacian operator come out from that. People talk about this already. So here, this Laplacian operator is associated with the filtration. So that's in our work. So this way, you give you a very powerful representation of biology. So basically, I finished differential geometry, algebraic topology. I talk a little about algebraic graph theory. And this is also very interesting stuff. So if you think about it, there's a molecular over there. I wanted to do the subgraph theory and pick up a certain type of atom instead of everybody. And from there, I have a adjacent matrix and also Laplacian matrix. And from here, we ask a question, come and heard the shape of drum? I mean, certainly that's a famous question asked like in the 70s, the way conclusion said you cannot. But actually, if you do some trick, you can do that. So what's the trick? If you include the filtration, you can do that. I'm going to get back to that a little bit later. So also we can use the geometric graph theory. So use centrality, things like that to avoid to the computation of the matrix. And then we can use the graphic theory to represent like a HLV. So people, if you use the original graph theory, use a eigenvalue, eigenfunction, it takes 120 years to do the job. But if you use the geometric graphic theory representation, you can get it done in something like a 20 seconds, or less than 30 seconds for this point here. This is a huge is a HLV. So that's a reduced complexity of computation. So the last thing I want to talk about, the mathematical part, is about the persistence spectrum graph, and also called persistence Laplacian. So we are going to make a use of spectrum graph theory, combinatorial graph theory. This is all the stuff. You go from a simplicial complex and K-chain and chain group, things like that, boundary group. And then you stop there. You stop at a boundary group, you don't go any further. You have an adjacent boundary operator. And then you have a Q combinatorial Laplacian operator. And this is a lot of people did work, include Professor Yost over there. So, and these things give you the Betti number, okay? So it's getting out by this equation. So what we did, I mean, this is everything so far. I'm looking to what we said here. We introduce a persistency over there, filtration. So we introduce the filtration to such a spectrum graph theory or a combinatorial graph theory. And then what we have is we end up a multi-scale spectrum graph theory. And we actually have a software for that, Hermes. So for this persistent spectrum graph theory. I just want to say this whole thing is a little bit more powerful than persistent homology. Why is that? Because it has all information of persistent homology past the spectrum theory. So you have a persistent spectrum analysis past the original barcode representation. So that's why it's much more powerful. So basically, I finished my mathematical introduction. So we use algebraic topology, differential geometry, graph theory, and we put everything into deep learning and tried to understand what is the major problem in the biology and tried to use this formulation for the scientific discovery. So I just want to show you two examples. One example I would like to say is there's a community-wide, worldwide competition, so-called drug design data resource D3R grand challenge. So it's about a computer-aided drug discovery type of competition globally in all kinds of different countries in the US and Europe and in other places. So this is what's going on for quite many years. So this competition, what it does, they give you just a sequence. So the protein sequence past the drug is not a real drug structure because this is not a three-dimensional structure. And what are they looking for? They're looking three-dimensional drug post, how this drug is interacting with this protein. And then they want to ask the binding energy. So all the drug has to bind it to the target, a protein over there, otherwise the drug doesn't work. So they ask you to do this prediction. They typically give you 2 or 3 this type of protein and then give you hundreds of this small molecular and ask you to do this prediction. And they have experimental data, but they don't tell you. And then what we did, we actually use those things, mathematics, featureization, put it into generative network. From there, we got a 3-dimensional structure and also with the drug over there. And from there, we further use the deep learning. This is a deep learning system. And further use the deep learning to predict the post and predict the binding affinity. So that's the competition we participated. And this is the result we have. I just want to say there are hundreds of people participated in this competition. And this is a lot of smart people over there because drug discovery is so important. A lot of big minds, they spend their lifetime on these things. So this is what we did. We did first, we got a few goals over there. That means goals and solve things like that. Out of 7 different categories. Second year, we got a lot more. And the third time we participated, for everything we participated, we actually get a lot of medals over there. So this work was done by, he has a biophysics background, otherwise Zixian and Dak, they just mathematicians. So he joined late, so he just participated in the last one. So I just want to say mathematician can do a lot. And the last thing I want to spend my time talk about is about correlating. I promised at the beginning, so what a mathematician can do about for correlating. So what a math can contribute over there. You look at it, this is the life cycle of the virus in the host cell. They start with here and then they go inside and release some genetic material, synthesize some proteins, things like that. They assemble other virus and then get out, try to infect other people. So that's a life process. It's very complicated because it's so complicated. So people get confused what's really looking for. Just for the rest of my talk, I wanted to say this is important. This is the so-called spike protein here. This is spike protein. And the spike protein interacts with the ACE2. This is the human cell, right? This is the human cell. So this is the inside human cell domain. This is the blue part is the human cell. So this is a human receptor. So this receptor is expressed in our lung and other place. So this one is interacting with the spike protein, and this interaction need this virus to get inside our cell. If we don't have this receptor, this without this protein, we will be safe, right? So, but unfortunately, this protein is also important for us, like control our blood pressure. So this interaction is crucial. So that's what we try to do. We try to, at even very early days, we realized that this is probably the most important part, is the protein-protein interaction. So we focus on these things. I will tell you what's our results. And then we certainly have a lot of other papers about this other stuff, but it turns out this is the part all the things we need to pay attention, protein-protein interaction. And then you look at the mutation. This is a horrible picture about a mutation. We got like 1.5 million people, their virus sample over there. And then we have a look at the mutation is 28,000 of mutations. And this each point is one mutation point. It's horrible for this whole virus. So many mutations, like this part, we have a few thousands of mutations on a spike protein. That's the one I'm talking about. So we are going to focus about. So there are 5,000-6,000 of mutation here. So what do we do? So the question is, what govern the virus transmission and evolution? So what is happening? What is going to happen in the future? So that's a question to ask. That means how this mutation happened and how they are going to affect us, right? So that's the question we try to answer. So I just want to show you one paper. This is a paper published last year. You see that. And this is a paper where basically say mutation strengthens the SARS-CoV-2 infectivity. This title tells the mutation, what the mutation is going to do. So we basically discovered mechanism of virus transmission and evolution more than 1 year ago. So basically we use algebraic topology, I want to say here. And then we also tried to, we revealed they become more infectious, and also they become more infectious is basically become a fundamental mechanism over there. I want to get into such a point in a few minutes. And we also look at all possible mutation at the virus, the place so-called is the spike protein receptor binding domain is a part of the spike protein. Spike protein is bigger, but the receptor binding domain is 10 times smaller. It's the place, the interaction between the virus and the ACE2, right? It's a protein-protein interaction site. So that's called a receptor binding domain. It's about roughly, it's 196 residues over there, right? So so from that residues, you can calculate it. There are 3,600 possible mutations. Among those possible mutations, we actually find a subclass is just 1,149 different possible most likely mutations. So far, we observed about 700 mutations. All of those 700 mutations are within our prediction, so-called most likely future mutation. And also in that paper, we point out two sites. One is the 455, other one is the 501. Those sites, they have a high chances to mutate into significantly more infectious covalent strains. So that's something we conclude use algebraic topology over 1 year ago. So this is what happened now. You see, we look at all the different most important variants over there, like alpha, beta, gamma, delta, and lambda, whatever. They all involve those two sites we mentioned over there, right? 501, 501, 501, 455, 455, 455. So this is something we basically remember. There are over 3,000 mutations over there, and also over 190 different sites. We point out those pinpoint those two sites, and this is what happened. Is just what we predicted. So, and also in the same paper, we said we have a hypothesis. We said we hypothesize the natural selection. We point out the mechanism, natural selection, favor those mutations that enhance the virus transmission. And if our prediction is correct, the predicted infectivity strengthened mutation will outpace the predicted infective weakened mutation. 1 year ago, we don't have so many mutations. We just got 89 mutations. When our paper first submitted, just 50 mutations. Finalized stage of our paper is 89 mutations, and we are able to find out the molecular mechanism of transmission and evolution. So this is mutations make the virus more infectious. So their populations grow. This is the frequency. And then the blue ones that make whereas less infectious, their population is low. So this is something we actually very proud of. We found this at the time. We don't have so much data. We actually get it right. So basically, at this point, as I said, there are thousands of mutations, tens of thousands of mutations over there. The more experiment, all the experiments, if done correctly, confirm our hypothesis. So basically, we know what is going to happen. So this is the other picture I show. Look at it. I said we have a 700 mutations on RBD, right? On the RBD. This is the RBD, this is the ACE2, their interaction over there. 7,000 mutations over there we observed at this point. Just look at the top 100 mutations. What I mean top means the ones they have a highest frequency, right? So they have a high frequency. This is the frequency. The red ones means high frequency. Top 100 high frequency mutations over there. So basically, all of them essentially, and every one actually is have energy higher than the average possible energy. Average possible energy is negative 0.3, and all for here you see the energy is so basically all the mutation top mutations there they strengthen the virus infection. And what is the chance for this to be how many odds, what's the odds for this to be accidentally we get it right? The accidentally get it right, the chance is just one chance in 1.2 nonlinear. So it's basically 10 to the 30. You can calculate, right? This is 100, so 1 over half, because you have a half probability of above the average energy, and half the probability lower the above energy. So you give you this number, okay? So this is how we did it. We got from people get a virus, and then we got a genome typing. We do genome, so genome analysis, and then we do protein-protein interaction. So about 700 different mutations we put over there, and we incorporate 130 antibody. And then go for topology barcode, and then deep learning, and then we predict everything. So that's what we did. And our prediction is basically as good as experiment. You see, this is our predicted DP mutation. This is experiment one. This is our predicted one. So that's what is happened. So for this type of problem. And our prediction for the ELA-Lini antibody, that was somehow originally was used by government, can be used now, they say, because of mutation, because those type of mutation, because disrupted antibody, like efficacy, like 25 times, things like that, they are no longer being used on the market. So this is our prediction. This is basically my last slide for the science part. You see, this is a lot of information. I call it the authors of imaging variants. That shows you the future, what is going to happen. So we have a alpha is not here. Alpha is a simple mutation. We have a beta, we have a delta, and we have a gamma, we have a lambda. So what is going on? The height over here is the total binding free energy change. The higher of this one, the higher the infinity, right? The higher infectivity. So you see delta is very high infectivity, and so far the most known mutation over there, variance over there, delta is the one has the highest infectivity because their binding free energy change is bigger. It's about 4.5 times more infectious than the original virus. And then you look at other things, and lambda is almost as infectious as a delta, but there's on top, there's a number, 59. What I mean by 59? Remember I said we use 130 antibody. We look at each mutation, how they're going to affect 130 antibody. If they're going to disrupt like a 59 antibody interaction with the virus, we put a number as 59, you see? So delta is going to disrupt 40, lambda is going to disrupt the 50 line. So that means lambda is going to be more dangerous. It's as infectious as delta, but it's going to destroy a lot of antibodies in people's body. So that's going to be this. So this lambda is going to be cause a lot of more trouble. And also you have a things called delta top. And here the y-axis is 1.5. Here is 2.5 here, right? So we have a lot of like a delta path. So the binding energy change is about 1.7, and it's going to destroy 82. Okay, 82. And also beta is going to destroy 90. So that's beta is really a vaccine breakthrough mutation. And the gamma is here, it's 81. And the infective, this one is high. And I put a few more other things there. So the beta path is going to basically destroy all kinds of antibody inside of. So that's beta path is more dangerous. And then we put other three, we just put other number like a mu and chi, and this things we just put over there. It's a lot of official names over there. And then you see they have a very high infectivity, and also they are going to destroy a lot of antibody. So those are the very dangerous things to come in the future. So basically, I wanted to summarize what we have been doing. I said everybody knows biology is so important because in the morning we have so many talks about biology. So the last frontier of science is biology. That is true. We have taken us so long to figure out every detail about this pandemic. That means we are our understanding of life and also a small things like a virus is still not there. I mean, we take it like 50, maybe 100 more years to figure out everything about biology. So that's why last frontier of science is biology. However, in my opinion, last frontier of biology is mathematics. So mathematics play a huge role in the biology. So we can have all kinds of different mathematics, excuse me, like different topology, different geometry, and different analysis, different graph theory, and other things, stochastic statistics, probability, differential equation, and also number theory, whatever. All kinds of things they can be used and also have a great advantage to be used for the biological science. So that's basically what I would like to say. Actually, I'm doing quite well in terms of timing. I got 11 minutes for people to ask a question. Yeah. Thank you very much. Yeah.
Yes. Thank you very much for this very rich talk with many biological examples. I suppose there are many questions. And again, if you want to ask questions, just type a Q into the chat, that suffices. Okay, the first question is by Guxing. Please go ahead and ask your question.
Thank you. Hello, Professor Wei, and thank you for your talk. So my question, or more like a comment, is about the precision homology. So in your slides, I noticed that the coefficients are all taken to be Z-mod 2. And I understand that's probably the computationally least expensive option. However, homology inherently carries torsion information, I mean, in integral coefficients. So let's say if we compute mod 2 homology and then mod 3 homology, and we compare the Betti numbers in a given dimension, and let's say the mod 3 Betti number is greater than the mod 2 Betti number, then we know that the integral cohomology has non-trivial torsions. But do you think that's significant information in your perspective?
At this point, we actually just use quite a simple things over there. Instead of getting into a lot more detail. But I'm quite sure because biology providing a test bed for all kinds of different mathematics. I'm quite sure those type of torsion or other information can make a difference over there, because biology, there's basically the whole protein or things like that. They don't have any symmetry or things like that. You need to find out that there are different features over there. All kinds of mathematics, they actually play a very important role. The detail is, I mean, what we need to do is try to figure out how to use it. That's it. Yeah, I'm quite sure the things can be, yeah, all kinds of mathematics. The more you look into mathematics, and the more you find out is important in the biological science. So that's, yeah, what I want to say. Yeah.
Okay. Yeah, thanks.
Okay, then let me also ask some questions. So in the beginning for the protein folding, if I understand correctly, you were just looking at the final shape when the protein was folded. And then the question is, for example, to distinguish the alpha helices or identify the alpha helices in the beta sheets. But would the data also allow you to look at the folding process itself and to trace how the homology is changing during the folding process from the linear string to the final folded shape?
Yeah, I think this is an excellent question. That's true. We actually tried to understand the interaction. That's why, because the protein folding process at each step involve a lot of interactions. So we tried to use all kinds of mathematical description, but the interaction associated with very high dimensional space representation. And we tried to use persistent homology to simplify such an interaction. So that's what we try to do. And to understand at each step, what is the interaction, the most dominant interaction associated with the transition state. And also final shape is just one thing. And certainly we in terms of folding problem, we look at the final so-called the minimum energy state. But the process involves something else. And also it's very important.
Okay, then good. Let me ask you another, maybe a little provocative question. So you are pretty good in predicting the future of the COVID-19 virus in terms of mutations or so. Could you also run your algorithm backwards and contribute to tracing the origin of the virus? Because that is, of course, a very controversial subject.
Wait, I know there's I mean, I don't know how to use the set of things we are doing to trace the origin, but we do. We have a paper. We are in the writing. We look at how the human ACE2 and interacts with various spike protein, and also we compare this with the bat ACE2 and the interaction with the spike protein, and also we compare with the dog and the cat. We have a list of different animals there. So basically information we got is the virus is already essentially optimized in all different animals. So certainly they already optimized with the human, but also they already optimized with a lot of different animals, like a bat and a dog and a cat. So we're actually writing a paper about what is the delta variant and of alpha beta, what is the impact of those variants to the dog. So we found out actually they are going to have quite a lot of impact with the dog. So be careful, just don't hold your dog if you have something. It's very dangerous because they can propagate to dog, and then dog involve a lot of set of variants over there, and then they transmit back to human. So that's things actually very dangerous over there. We can study that because the protein-protein interaction, that's basically what's going on. Yeah.
Okay. And you mentioned also that you look at the spectra of the Hodge Laplacian in addition to the topology. So what additional information do you get from that when you look at biological data?
We have a code for Hodge Laplacian. We have code for this, but we actually so busy in the past year and a half with correlating. So you realize we spent basically most people in my group so work on those type of, because it's the emergency problem. So we haven't done too much application of the evolutionary de Rham-Hodge theory for biology. Except we have a collaborator in chemistry, he's writing a paper about that. But for my own group, I haven't done much use this algorithm for biology because we are just too busy. We got a pressure to answer those emergency problems in the virus. So yeah, but I'm quite sure this is going to be more powerful than Poisson homology. We did everything we did so far is use Poisson homology. Yeah, because the software is also thank the community that develop a lot of software over there. It's very convenient to use those Poisson homology things to do biology at this point. So include like we did in the many years we did code for a lot of Poisson homology analysis. So this is make a whole thing fly. I mean, this is why in my picture I said not only big guys over there, right? So yeah, why this one doesn't show up? Yeah, and also include the whole community. Not this one. Okay, I see. It's the earlier one. I have those type of things. So the community, they are really providing the developments, and we are at a point we can use those advanced mathematics for biological science, right?
Okay, then thank you very much. I don't see any further queue in the chat. And so that means that we can reach the end precisely on time of today's session. And I would like to extend my cordial thanks to all speakers for giving very interesting talks and providing us a lot of material and ideas to think about further. And I'm looking forward to the next 2 days of this conference. So thank you, everybody, and have